Hello champs, I hope you all are doing great. My name is Biswajit, I'm your botany teacher. Today we are going to discuss DNA fingerprinting and ecosystem. And in ecosystem chapter we will discuss what is ecosystem, what are the different components of ecosystem, structural aspect of ecosystem, functional aspect of ecosystem, ecological succession, etc. in detail. Okay, so let's start with the DNA fingerprinting. So the very first thing which you should remember here is who was the person to develop this technique? It was Alex Jeffrey. Important. And this DNA fingerprinting is based on two different things. One is VNTR, second is RFLP. So what do you understand by VNTR and RFLP? Let us try to understand in detail. Okay, so the DNA which is present in a body, or cell or chromosome can be of three different types. One is non-repetitive DNA, second is tandem repetitive DNA, and third is dispersed repetitive DNA. Okay, so what do you understand by this? Let us try to understand with the help of these diagrams. So suppose these are DNA molecules. Okay, and here A, B, C, D, these are some sequences, base pair sequences. Okay, so in the first case you can see not a single sequence is repeated. So this type of DNA is non-repetitive DNA. In second case, sequence A is repeated. In third case also sequence A is repeated. So these two DNA are called as repetitive DNA. But in second case, the repeating unit A is repeated, is present continuously. And if the repeating unit is present continuously, that will be called as tandem repetitive DNA. And if the repeating unit is present discontinuously, that will be called as dispersed repetitive DNA. Okay, so here I have taken some repetitive DNA molecules. Okay, so these are repetitive DNA. RU stands for repeating unit, and in all these cases you can clearly see that the repeating unit is present continuously. So all these are called, all these DNA will be called as tandem repetitive DNA. Okay, and there is a difference in the and these DNA molecules show some variations. Variation in what? The number of repeating unit. And because these are tandemly repetitive DNA, so I will call this DNA as TR, tandem repetitive DNA. And because these tandem repetitive DNA molecules, they show variation in their number of repeating unit. They show variation in the number of repeating unit. That is why I will call this DNA molecules as VNTR, variable number tandem repetitive DNA. Okay, and what about the repeating unit of the VNTR? The repeating unit of the VNTR shows differences or variation in its number. Okay, and the number of the repeating unit of the VNTR varies from one chromosome to another chromosome of the same individual, or I can say among the different individuals. But the sequence of the repeating unit of VNTR is always same in a species, and that is why I will say that the sequence of repeating unit of VNTR is conserved. I repeat, in VNTR I take a repeating unit, and the repeating unit shows variation in its number, but shows conservation in its sequence. Okay, and the variation variation in the number is present among the different individuals of same species and among the different chromosomes of the same individual. Okay, now the VNTR is an example of satellite DNA, and satellite is of two types mainly. One is mini satellite, another is microsatellite, and this is an example of mini satellite. And this VNTR is inheritable. That means the VNTR of a father or mother will be transmitted to the children, and that is why we use VNTR, or that is why we use DNA fingerprinting to solve paternity cases or maternity cases. Why? Because VNTR is the basis of DNA fingerprinting, and VNTR is inheritable. That is why we use DNA fingerprinting to solve paternity case or maternity case, paternity maternity dispute cases. Okay, now what about the RFLP then? RFLP, let us try to understand with the help of these diagrams. Okay, RFLP is basically restriction fragment length polymorphism. Okay, so these are DNA molecules. Okay, and the DNA will be present in the chromosome. So suppose these are chromosome 5, all are chromosome 5. Okay, so here this is VNTR, this is VNTR, and this is VNTR. And on either side of the VNTR, I will have some sequences, and these sequences are called as restriction sites. Okay, and these restriction sites will be recognized and cut by restriction endonuclease. Okay, so restriction endonuclease will come and cut these DNA molecules. Okay, and once the DNA molecules are cut by restriction endonuclease, I will get fragments. So I will get fragment 1, 2, 3, fragment 1, 2, 3, fragment 1, 2, 3. And you will see that the fragment 1 will be same length. Fragment 3 will be also same length. And this is from, suppose this is chromosome 5 from person A. This is chromosome 5 from person B. And this is chromosome 5 from person C. Okay, so you will see that the fragment F1 obtained from person A, B, C will be same. Likewise, the fragment 3, F3 obtained from person A, B, and C will be same. But if I talk about the length, not sequence, I'm talking about the length. Okay, if I talk about the length of the fragment 2 obtained from person A, B, and C, they will vary from each other. So here I'm obtaining the fragments. I'm obtaining the fragments over here. Okay, so I'm talking about the fragments. Okay, I'm talking about the fragments, and these fragments are obtained after the cutting of the DNA molecules. Okay, and cutting by what? Cutting by the restriction endonuclease, and that is why these fragments are called as restriction fragments. That is why they are called as restriction fragments. Okay, and here I am talking about the length of these fragments. Okay, so I will call this as length, restriction fragment length, and you can see that the length of this fragment and the length of the fragment and the length of this fragment, they are varying. That means you can see that this VNTR has more than one length, and if VNTR has more than one length, I will call this as polymorphism. So this is restriction fragment length polymorphism. So the restriction fragments of the VNTR, which is obtained due to restriction digestion of DNA molecules, they will show more than one type of length. That means they will show length polymorphism. Clear? So this is restriction fragment length polymorphism. And in DNA fingerprinting, we use a technique which is called as electrophoresis. So what do you mean by electrophoresis? Electrophoresis is nothing but a technique of separation. And in this separation technique, we use we separate the charged particles. We separate the charged particle under electric field in the gel. Okay, so in gel under electric field, we separate charged particles. Okay, so if I want to separate the DNA molecules, then I will use agarose gel electrophoresis. Which type of gel we use to separate the DNA fragments? Under electric field, we use agarose gel, and that type of electrophoresis will be called as agarose gel electrophoresis. Okay, and when so when do I separate the DNA molecules? Okay, once I do the restriction digestion. So first of all, I will collect the DNA sample. Okay, then I will digest the DNA molecules with the restriction endonuclease. So I will get the DNA fragments of different length. Okay, and then I will load the DNA molecules and in the gel, and I will allow the electrophoresis to run. Okay, so now the separation of the DNA fragments will be decided by the size of the DNA fragments. Okay, DNA fragments. So suppose here you can see that this is the gel electrophoresis setup. Okay, and this part is basically gel, and you can see here clearly that this gel is having some depressions, and in these depressions I will load the DNA sample. Okay, and the DNA sample will have a number of DNA fragments of different length. Okay, and we know that the DNA has negative charge. So if I supply the power, then what will happen? The negatively charged DNA will move towards the positive electrode. And as I said, the DNA sample has DNA fragments of different lengths. That is why the DNA fragments of different length will move at different paces. So the smallest DNA fragment will move fastest and the largest DNA fragment will move slowest. Okay, and after certain time, you can see the DNA band like this. So suppose this is one DNA band, another DNA band, another DNA band, another DNA band. So suppose here I'm taking 4 DNA bands. Okay, so this DNA band will have a number of DNA molecules, but all the DNA molecules present in this band will be of same size. Here also a number of DNA molecules are present, but in this case all the DNA molecules will be having same length. Okay, and this will be the shortest DNA fragment and this will be the largest DNA fragment. Here also I will get the DNA band like this, like this. Okay, so I'm getting the DNA band over here. Okay, so the I can say smaller is the DNA fragment, fastest it will move. Okay, so this is the point you have to remember over here. So any separation of the charged particle under electric field in the gel is called as electrophoresis. We can separate DNA molecule by electrophoresis, RNA molecule and protein also. We can separate them. And when we separate DNA molecules, we use which type of gel? We use agarose gel. That is why we call that electrophoresis as agarose gel electrophoresis. And in this case, the separation of the DNA fragments is based on the size of the fragments. Okay. Now, once we perform the electrophoresis, now we are going to perform the blotting. So what do you understand by blotting? Blotting is nothing but the transfer process. Transfer of what? Transfer of substances present in the gel to the membrane. And the membrane which is used in the blotting is called as blotting membrane. It can be nitrocellulose membrane, it can be nylon membrane. Okay, so we have done the separation by electrophoresis. So in the gel I have either DNA or RNA or protein. And in DNA fingerprinting, I will talk about DNA only. So in the gel, agarose gel, I have DNA bands or DNA fragments. Now I want to transfer the DNA fragments to a membrane. Okay, and that transfer of DNA fragments from gel to a membrane is called as Southern blotting. Why? Because this technique of blotting in which DNA was transferred to membrane was first done by Southern scientists. The name of this scientist was Southern. That is why this technique is called as Southern blotting. Okay, and if I transfer RNA from gel to the membrane, that is Northern blotting, and if I transfer protein from the membrane from the gel to the membrane, that is known as Western blotting. Okay, so this is blotting. So when I perform Southern blotting, so what does happen in Southern blotting? Let me explain you. So this is alkaline solution. And this alkaline solution has the property or ability to denature the DNA. That means it will separate the double-stranded DNA to form single-stranded DNA molecules. Okay, and in the alkaline solution, I have taken a filter paper. Above the filter paper, I have taken the agarose gel, which will be having the DNA fragments. Okay, and the DNA fragments present in the agarose gel, they are double-stranded in nature. Above it, I will keep the nitrocellulose membrane, and above it, I will again keep the filter paper, and above that I will keep some weight. So when I leave this setup as it is, as such, for some time, what will happen? Because of capillarity, the alkaline solution will move in upward direction. And when the alkaline solution moves in upward direction, it will cross the filter paper, then it will cross the agarose gel. And when the alkaline solution crosses the agarose gel, it will carry the DNA molecules present in the agarose gel along with it, and it will come to the nitrocellulose membrane. Now the solution will cross the nitrocellulose membrane, but the DNA cannot cross the nitrocellulose membrane. So the DNA which was carried by the DNA fragments which were carried by the solution will remain attached to the nitrocellulose membrane. Okay, and when the alkaline solution carries the DNA molecules from the gel, so during the process of this transportation, the alkaline solution will cause the separation of the two strands of a DNA. Okay, that means suppose here this is gel is there, okay, and DNA molecules are present, okay, so the double strand DNA is present. So this is one strand, this is another strand. So this is double stranded DNA, okay, present in gel. So alkaline solution will carry this one in upward direction. So this is moving in upward direction, and when it moves in upward direction, it will separate. Because of what? Because of alkaline solution, and it will move, and here there will be nitrocellulose membrane, and on the nitrocellulose membrane the single strand DNA molecule will bind. Here also, suppose one double strand DNA, suppose DNA band are there, okay, so double strand DNA is present in the gel, so they will move in upward direction by alkaline solution, and when it moves, they will separate from each other and they will bind to the nitrocellulose membrane. And that is why the DNA present in the gel is double stranded, but in present in the membrane is single stranded. Clear? Good. So this is blotting. Blotting is nothing but the transfer of substances from the gel, electrophoresis gel, to the membrane. What type of substance you transfer? Based on that we classify that. If you transfer DNA, southern blotting. If you transfer RNA, northern blotting. If you transfer protein, that is western blotting. And the membrane which we use is called as blotting membrane. It can be nitrocellulose membrane or it can be nylon membrane. Clear? Good. Now once we perform blotting, what we are going to do, we will separate the nitrocellulose membrane, okay, which will be having the attached single stranded DNA fragments. Now we will add the probes designed against VNTR to the nitrocellulose membrane. Okay, so some of the probes, they will go and bind to those DNA molecules which have VNTR, which are basically VNTR. Okay, and then I will wash the membrane so that the unbound probe will get detached. Okay, then I will allow the membrane to get dried, and once it becomes dry, I will keep the extra film above it and I will perform extracrystallography, and the DNA binding pattern will be observed on the extra film. Okay, and the DNA binding pattern which I obtain on the extra film will correspond only to the VNTR, not other DNA fragments. In gel or in nitrocellulose membrane, a number of DNA fragments are bound, but only those fragments will be those bands will be present in the extra film, which correspond to the VNTR, Okay, and by looking at the DNA binding pattern on the extra film, we can compare. We can say okay, this DNA is from this person and this DNA is from this person. Okay, so what are the various steps we use in DNA fingerprinting? The very first step is we isolate the DNA from sample. Okay, once we isolate the DNA, we perform restriction digestion by restriction endonuclease. So I will get a number of fragments of DNA molecules. Then I will do the electrophoresis to separate the DNA fragments. Then I will perform the blotting, and once I perform blotting, I will take out the blotting membrane. Okay, then I will hybridize the blotting membrane with the probe which is designed against the VNTR. Okay, then I will wash the nitrocellulose membrane to remove the unbound probes. Then I will allow the membrane to dry. Then I will keep the extra film above it, and I will allow extracrystallography, and then I will observe the DNA binding pattern. Okay, so this is what the various steps used in DNA fingerprinting. Clear? Great. So here a diagram is given here. Okay, so this is this is the well. These are the wells. Okay, and in these wells I have loaded some DNA sample. Okay, in the well C, I have loaded DNA sample which is obtained from the crime site. Okay, so from the crime site, the DNA which is obtained will be having VNTR also. Okay, so from the crime site, suppose the DNA which is obtained, suppose these are the chromosome 5, these are chromosome 2, and these are chromosome 16. So in chromosome 5, you can see the repeating unit is present continuously. Here also repeating unit is present continuously. That means this sequence is basically nothing but the VNTR. This is VNTR, this is VNTR, this is VNTR, this is VNTR, and this is VNTR. Okay, so first of all, once I isolate the DNA, I will digest the DNA by means of what? By means of by the help of restriction endonuclease, and then I will load that DNA into this well. Okay, now the DNA molecules will get separated. Okay, and as I have already told you, the smallest fragment will move fastest. Okay, so this one is the smallest one, which has only two repeating units, and that is why this one will be this VNTR. Okay, and here I'm having 5 repeating units, so this is this so this DNA band corresponds to this VNTR. Okay, so like this. So once I perform the, and here I load the DNA sample, fragmented DNA sample obtained from the person A, and here I load the DNA sample obtained from the person B. Okay, and once I perform the electrophoresis and then blotting and then hybridization and X-ray film, then I will compare the DNA bending pattern, and this DNA bending pattern are of VNTR only, then I will compare the DNA bending pattern. So you can clearly see that the DNA bending pattern of the VNTR of the obtained from the crime site is identical to that of the person B, and that is why I will say that, okay, the suspect A, among suspect A and B, which one is the criminal? It is the B. Clear? So this was about DNA fingerprinting. So let me repeat, what are the various steps? The first step is isolation of DNA. Then the next step is restriction, digestion, of DNA by restriction endonuclease. Then we perform the electrophoresis, gel electrophoresis. Then we perform blotting. Which type of blotting? Southern blotting. Okay, and then we add the probe to hybridize. So I will say hybridize with probe which is designed for VNTR only. Then we perform X-ray crystallography. Okay, then compare the DNA bending pattern. Okay, so these are the various steps used in the DNA fingerprinting. So what are the significances? The significances are, this is used in forensic science. Okay, and this is used to solve paternity and maternity dispute cases. Maternity dispute cases. To solve this, we use DNA fingerprinting. Okay, now let's talk about ecosystem then. So ecosystem is basically a self-sustaining or functional unit of living world. Okay, and this ecosystem can be classified, just a minute, let me check the time. Okay, this ecosystem can be classified based on the place where they are present or based on the involvement of human being. If they are present on terrestrial habitat, we call that as terrestrial ecosystems. Examples: forest, grassland, desert, etc. And if they are present in water body, we call that as aquatic ecosystem. Example: pond, lake, river, wetland, sea, estuaries. And if an ecosystem has developed without any involvement of human being, that is natural ecosystem. Examples you can see here: forest, marine ecosystem. And if an ecosystem is developed by human being, that is anthropogenic or man-made or artificial ecosystem. And some of the examples you can see: crop fields, garden, aquarium, agriculture. Okay, agriculture fields. These are man-made ecosystems. And among man-made ecosystem, the largest one is the agro ecosystem or agriculture ecosystem. This is the largest man-made ecosystem. And when I compare natural and man-made ecosystems, so natural ecosystem is better than man-made ecosystem, except with respect to productivity. Okay, so what are the features of anthropogenic or man-made ecosystem. Man-made ecosystem has no self-regulatory mechanism. Natural ecosystem has self-regulatory mechanism, but man-made ecosystem has no self-regulatory mechanism. Second, in man-made ecosystem, simple food chains are found, but in natural ecosystem, complex intervened food web is present. In case of anthropogenic ecosystem, little nutrient cycle takes place, but in case of natural ecosystem, the nutrient cycling is very high. And in man-made ecosystem, diversity is very less, but in natural, diversity is high. Okay, in man-made ecosystem, productivity is high. This is important, and natural less as compared to artificial. Okay, so only with respect to productivity, this anthropogenic or man-made ecosystem is better as compared to natural ecosystem. Otherwise, natural ecosystem is better than anthropogenic ecosystem with respect to other criteria. Okay, now, so what are the different components of ecosystem then? What are the different components of ecosystem? Ecosystem has two different components. What are those? One is abiotic component and second one is biotic component. So what do you mean by abiotic component? Any component of ecosystem which is non-living, that will be abiotic component. And the abiotic components can further be classified into climatic factors, okay, or edaphic factors or topographic factors. So what do you mean by climatic factors? The climatic factors includes temperature, water, light, wind, humidity, air, currents, etc. Okay, and the edaphic factors are related to soil. So any factor which is related to soil, that will be edaphic factors. Okay, so those factors which are related to soil structure or soil composition, they will be coming under the edaphic factors. And any factor which is related to the physical feature of earth, they will be called as topographic factors. Examples: slope, valley, mountain, plain, etc., etc. Okay, so these are abiotic components of what? Ecosystem. And apart from abiotic factors, I will also have biotic components in ecosystem. And the biotic components will be having life. They can belong to plants, animals, or microbes. And the microbes, in microbe I can include Monera, Protista, and fungi. Okay, and the biotic components are further classified into various groups like producers, consumers, okay, detrivores, parasites, etc., etc. So let us talk about the producers first. So what do you mean by producers? Those organisms which can produce their own food by the process of photosynthesis. So there will be producers. So all the green plants, they come under the producers. And because they are able to use inorganic carbon, that is CO2, that is why they are called as autotrophs. And because they convert one form of energy to another form of energy, that is why they are called as converters or transducers. We know the producers, they perform photosynthesis, and during the process of photosynthesis, the light energy is converted into chemical energy. That means here one form of energy is converted into another form of energy. That is why they are called as converter or transducers. And so we know what is producers. Green plants, photosynthetic plants, they are producers. So do you think that in different ecosystems the producers will be same? No. The nature of producers will differ according to the ecosystem. Okay, so if I talk about the terrestrial ecosystem, different types of producers will be there, but the main producers in terrestrial ecosystem will be herbaceous and woody plants. Okay, likewise in aquatic ecosystem, different types of producers will be there, but the main, or I can say the chief producers in aquatic ecosystems will be phytoplanktons, algae, floating and margin plants. Okay, and in 11th class there is a sentence in diatoms that diatoms are the chief producers of ocean. Okay, now let us talk about the consumers then. Consumers are those organisms which cannot produce their own food. That means they cannot use inorganic carbon, and that is why they depend on the produces either directly or indirectly, okay, for food. And this and they are because they are not able to use inorganic carbon, so that is why they are called as heterotroph. And these consumers are further classified into various groups like primary consumer, secondary consumer, tertiary consumer, top carnivore, etc., etc. Okay, so what do you mean by primary consumer? Those organisms which depend on the producers directly, they are called as primary producers. And they are also called as the primary consumers, they are also called as first order consumers. And because they derive their nourishment from the plant or plant products, that is why they are called as herbivores, and they are called as key industry animal. Why key industry animal? Because these consumers, they convert the plant material to the animal material, plant substance to animal substance, that is why they are called as key industry animals. Okay, and the examples of the primary consumers in terrestrial ecosystem I can say grasshopper, cow, deer. In aquatic ecosystem I can take mollusks, tadpole, mosquito larva, etc., etc. Okay, now what about secondary consumer? The secondary consumer are those consumers which feed on primary consumer, and they are called as second order consumer or primary carnivore. Okay, in terrestrial ecosystem, some of the examples of secondary consumer are toad, spider, lizard, centipedes, insectivorous birds. Okay, and in aquatic ecosystem I can take the example of hydra, okay, frogs, small fishes. And what about tertiary consumer? These are those consumers which feed on the secondary consumer. They are also called as third order consumer or secondary carnivores. Okay, and in terrestrial ecosystem the example I can take is snake, and in aquatic ecosystem I take the example like large fishes. Now the other consumer is top carnivore. These are those consumers which are not eaten by any other organism. Okay, they may belong to the primary, secondary, or tertiary carnivore, no problem. And the examples of the top carnivores are tiger, lion, panthers, falcon, peacock. Okay, now what about the other component? So far we have discussed about two biotic components. One producers, second consumer. Now we are going to discuss about third component or third biotic component, and that is the decomposers. And these decomposers, they break down the large complex organic molecule into small simple inorganic molecules, and that is why they are called as reducers. And they are always microbial in nature. They may belong to fungi, bacteria, or flagellates. Okay, and they are responsible for performing nutrient cycling. Okay, so what do the decomposers do? So they will decompose the organic matter. Okay, and the organic matter which is decomposed is basically called as detritus, and the detritus is basically complex organic matter. So I will have the complex organic matter. Okay, and this complex organic matter will be first broken down into simple organic matter. Okay, and from the simple organic matter the inorganic matter compounds will be released. So this is the process of what? Decomposition. This is the process of decomposition. Okay, so this is the process of decomposition. Decomposition. Okay, so what are the functions of decomposers in an ecosystem? They act as natural scavengers, and they are responsible for performing they are responsible for nutrient cycling. Okay, now the fourth biotic component is scavenger. These are those organisms which feed on dead or injured animals. The examples you can see: carrion, beetles, okay, storks, crow, vultures, etc. What are detritivore? Those organisms which feed on detritus. What are detritus? Detritus are basically dead organic matter. That is detritus. It can be, I can take the example of what? Fallen leaves or twigs or dead animal body or human excreta, etc., etc. They will come under detritus. Okay, example is termite. Parasites are those organisms which obtain their nourishment from the food, so from the host. Okay, and the examples I can take is parasitic bacteria, parasitic fungi, or parasitic protozoa or worms. These are the examples of parasites. Now, so we are done with the biotic components. Okay, ecosystem has two different types of components, biotic and abiotic. Okay, biotic components are classified into various groups like producers, which produce their own food, consumers, which depend on producers for their food. Okay, consumer is of various types: primary consumer, secondary consumer, tertiary consumer, and top carnivore. Then we talked about parasites, detrivores, and scavengers. Okay, now let us talk about the structure of ecosystem. Ecosystem has two different aspects: structural aspect and functional aspect. So what are the structural aspects of ecosystem? One is species composition. One is species composition. Okay, and second is stratification. These are the structural aspect of ecosystem. So what do you understand by species composition of an ecosystem? So when I say species composition of an ecosystem, that means I'm trying to understand how many different types of species are there in an ecosystem, and what are the relative number of the or the number of each and every species of species in an ecosystem. Okay, so this is species composition, and the species composition varies from one ecosystem to another ecosystem. That means different ecosystems have different species composition. So maximum species composition is maximum or high in ecosystems like what, tropical rainforest. Okay, and I can say minimum in ecosystem like desert. Okay, now this species composition is a product of what? Two things. One is species diversity, one is species evenness, second is species richness. We will talk about that in biodiversity and conservation. Okay, now the second structural aspect of ecosystem is stratification. So what do you understand by stratification? Stratification is nothing but the vertical distribution of different species of an ecosystem. Okay, we are distributing the different species vertically in an ecosystem. Okay, so I have taken two examples over here. So in water body, we can see green algae, brown algae, and red algae. The different species of green algae, brown algae, and red algae. Okay, so suppose this is water body. Okay, and in the water body I will have water. Okay, so water is present. So you can clearly see that the green algae are the species of green algae, they are distributed on the upper layer, and the brown algae are present lower to the green algae, and the red algae is present both upper layer and lower layer, lowest layer. So I have distributed the species of green algae, brown algae, and red algae vertically, and this vertical distribution of these species of these members is called as stratification. So why green algae are present at the surface or at the upper layer? Because green algae can use only those wavelength of light which are found in the upper surface. Okay, and brown red algae, they are found in both upper surface and lower region. Why? Because they can use those wavelength of light which can penetrate and reach depth of the water body, or which have less penetrating power, or which are found at the surface of the water body. Okay, and that is why they are present at both surface as well as lower part of the water body. Okay, and if I take the example of what? If I take the example of forest. Okay, so in the forest, the top layer will have trees, second layer will have shrubs, and bottom layer will have the grasses or herbs. Okay, so let me draw the diagram here so that you can understand. So these are the trees. These are the trees. These are the trees. Okay, this is the first or top layer. Okay, then the second layer will be there. Okay, so let me draw the second layer over here. This is second layer and this is third layer. So you can clearly see that there is a vertical distribution. Okay, so and this is nothing but the stratification. Okay, now Let us talk about the functional aspect of ecosystem. Functional aspect of ecosystem. The functional aspect of ecosystem includes productivity, decomposition, energy flow, and nutrient cycling. These are the four functional aspect of ecosystem. Okay, so what do you mean by productivity? So before understanding productivity, you should know what is production. You should know what is production. Okay, so what do you mean by production? Production is basically is formation or synthesis of biomass per unit area per unit time. Okay, not per unit area, per unit time. Per unit time, how much biomass is produced, that is production. So I can say here that the biomass produced per unit area. How much biomass is produced per unit area, that is called as production. Okay, and what is productivity? That is the rate of biomass production. That means this is the biomass produced per unit area per unit time. So I can say productivity is basically biomass produced. How much biomass is produced per unit area per unit time. So per unit time, how much biomass is produced, that is called as productivity. Okay, or I can say rate of biomass production is known as productivity. And the productivity of different ecosystem varies. Okay, so maximum productivity is observed in ecosystems like coral reef, tropical rainforest, sugarcane field. And minimum or least productivity is observed in ecosystems like desert, deep sea. Okay, now what is primary productivity? Productivity is of various types. Okay, one is primary productivity. So what do you understand by primary productivity? Primary productivity is basically the productivity due to the producers. That means how much biomass is produced by the plants by the process of photosynthesis per unit area per unit time. That is primary productivity. Okay, and this primary productivity has units. The units of primary productivity can be expressed in terms of weight or in terms of energy. In terms of weight, I can say what is the unit of primary productivity? I can say kg. Okay, this is the kg per unit area meter square per unit time, that is per year. Okay, in terms of energy, the unit of primary productivity or productivity will be kilocalorie per meter square per year. Kilocalorie per meter square per year. Okay, sorry, here I should write only gram, not kg. Okay, this is gram. Very sorry, gram per meter square per year. So this is primary productivity. This is the unit of primary productivity. And the primary productivity is again of two types, GPP and NPP. What is GPP? The amount of biomass produced by plants per unit area per unit time is primary productivity, gross primary productivity. And after respiration, how much biomass is left, that is known as net primary productivity. And that is why I can say that GPP minus respiration will be NPP. Okay, productivity is biomass production rate of biomass production by plant. If the productivity is due to plants, I will call that as primary productivity. And the biomass produced is called as synthesized is called as GPP. And after respiration, the left part is called as NPP. And this NPP is available to the consumers and the decomposers, not GPP. This is very, very important point. You will be asked which of the following is available to the decomposers, GPP, NPP, both. No, it is NPP, not GPP. Okay, some part of the GPP will be used by the plants. Okay, now the second type of productivity is secondary productivity. So what do you understand by secondary productivity? The productivity or the biomass produced per unit area per unit time by the consumers. That is secondary productivity. Okay, and the third type of productivity is community productivity. So the rate of biomass production by the community. by the community, that is called as community productivity. So rate of biomass production by plants, primary productivity, by consumers, community productivity, by secondary by consumers, secondary productivity, by community, that will be community productivity. Okay, now the second functional aspect of ecosystem is decomposition. Okay, so the second functional aspect is decomposition, and this decomposition is also very, very important, and it is in fact equally important as compared to the productivity. Okay, and do you think that the productivity is the decomposition is same at all places? No. The main site of the decomposition is upper layer of soil and bottom of water bodies. Okay, so the main site of decomposition are upper layer of soil and bottom of water bodies. And the substance which undergoes decomposition, or the raw material of decomposition, is basically detritus. Okay, so the raw material for decomposition is detritus. So what do you mean by detritus? Detritus is basically dead remains of organic matter. Okay, and the examples I can take is dead leaves, bark, flowers, or dead remains of animals, human excreta, etc., etc. Okay, now the decomposition is done by decomposer and other organisms also. So let us try to understand the mechanism of what? Decomposition. Okay, as I have told you that the decomposition starts with detritus. It starts with detritus, and this detritus is basically organic, and this is large and complex. This is large and complex organic molecules, right? And this detritus will be acted upon by some organisms, and those organisms will obviously be called as what? Detrivore. They will be called as detrivore. Okay, now the detrivore will cause the fragmentation of the detritus. Okay, now the detritus will be converted into fine particles. And this process is called as what? Fragmentation. What do you call this process? You call this process as fragmentation. This process will be called as fragmentation. Okay, and this fine particle has low surface area, high surface area upon volume ratio, and this detritus has low surface area upon volume ratio. Okay, and these fine particles are also organic in nature. These are also organic in nature. Okay, and because these fine particles have high surface area upon volume ratio, that is why they can be easily acted upon by the decomposers. Okay, so now these fine organic particles will be acted upon by what? By the decomposers. So in the second step, the decomposers play their role, not in the first step. Decomposers will come and play their role. Now these fine particles will be, which is basically organic, that will be converted into humus. And this process of formation of humus from the fine particle is called as humification. Okay, and this humus is again organic in nature. Okay, now this humus will be further acted upon by the decomposers. Okay, and from the humus the minerals will be released. The minerals, that is the inorganic substances, will be released. And because in this process the minerals are released, that is why this process will be called as mineralization. that will be called as mineralization. Okay, and these minerals, some of the minerals will be uptaken by the plants. Okay, that means that will enter into the biotic world, and some will be leached. Some will be leached. So I will say then the minerals will be they will be undergoing leaching. They will be undergoing leaching. So what are the different processes of involved in the process of decomposition? Fragmentation, humification, mineralization, and leaching. Okay, and during the process of fragmentation or humification, in these processes also some amount of minerals will be washed downward along with the percolating water. That means in this step also some amount of leaching will take place. In this step also some amount of leaching will take place. So I can write here some leaching will be taking over here, and some amount of leaching will also take place over here. Okay, so I can say the process of decomposition. The process of decomposition includes 4 different events: fragmentation, humification, mineralization, and leaching. And all these processes take place simultaneously. And the process of decomposition is not performed alone by the decomposers. No, it is also performed by the detrivores. In fact, the very first step of the decomposition is done by the detrivores. Okay, so what is the need of the detrivore? Because the detritus, which is the raw material of the decomposition for the decomposition, has low surface area upon volume ratio. So the digestive enzymes released from the decomposers, they cannot act upon them easily, and that is why they have to be small in size, so that the surface area upon volume ratio will be high, and so that the enzymes will act upon them easily. Clear? So this is the process of decomposition. This is the process of decomposition. Okay, now let us try to understand something more about the humus. Okay, so the humus is basically organic matter. Humus is basically organic matter, and it does not have any specific form. It is without any specific form. So that I call that as amorphous in nature. Okay, and this is organic, and this is dark in color. This is dark in color, and this is rich in cellulose, lignin, and tannins. Okay, and this is highly resistant. This is highly resistant to microbial action. Okay, and that is why the microbes will perform their activity. They will digest the humus very slowly, and that is because the humus is highly resistant against the microbes, and the decomposers are also microbes. That is why this step of mineralization is slower in nature. Okay, why? Because humus is highly resistant. So I can write here this process is very slow. This process is very slow. What is the reason? Because humus is highly resistant. Okay, next, that is why I have written that the humus undergoes decomposition at extremely slow rate. Okay, and this is slightly acidic. This is very, very important. Okay, this is not alkaline, this is acidic, but this is slightly acidic, and this is colloidal substance, and this is the reservoir of minerals. This is the reservoir of minerals or nutrients. Clear? So if and because humus is reservoir of nutrient, that is why if a soil is humus rich, that will be more fertile. If it is if the soil has less humus, that will be less fertile or sterile soil. Okay, now so we have learned what is decomposition. Okay, so let us try to understand what are the factors which affect the process of decomposition. So various factors affect the nature of decomposition, rate of decomposition. The first factor is the chemical nature of the detritus. Okay, so different types of detritus undergo decomposition at different rates. So if a detritus has high amount of lignin, chitin, or cellulose, that will undergo decomposition at very slow rate. But if a detritus has nitrogen rich compounds like proteins, nucleic acids, or water soluble carbohydrates like carbohydrates, they will undergo faster decomposition. That means the nature of the detritus, chemical nature of the detritus, is determining the rate of decomposition. The second factor is the temperature. Okay, so above 25 degrees Celsius, the rate of decomposition is high. That means I can say decomposition rate will be active high, or I can say active decomposition will take place. And below 10 degrees Celsius, the decomposition will be very, very slow. Very, very slow. The third factor which is going to affect the rate of decomposition is moisture. Okay, and optimum. So if the decomposers are provided optimum moisture, then decomposition rate will be high. Okay, so when I talk about the just before some time I said above 25 degrees Celsius, decomposition is active. So when I talk about the desert will be having more than 25 degrees Celsius, but still the rate of decomposition in the desert is low. Why? Because in that case moisture is very, very less. The condition is dry. Okay, so moisture is also important. Not only the temperature, moisture is also important, because in desert there is prolonged dryness. There is very less moisture. That is why the rate of decomposition is very, very slow. Okay, the fourth factor is aeration. Okay, so the aeration is required for the activity of decomposers and detritivores. Why? Because they have to produce energy in the form of ATP, and for that they have to respire. Okay, and for that they need oxygen. Okay, so decomposition is done by detritivore and decomposers, and to perform that they need energy. They need to be they need to survive, and for survival they need ATP, and for ATP they need oxygen or aeration. Okay, so no doubt the chemical nature, temperature, moisture, and the aeration, they are required. They affect the process of decomposition. But among these four, the most important one which are going to affect the rate of decomposition are temperature and moisture. These are the most important factors which affect decomposition. Very, very important. Okay, now let us try to understand one term which is nutrient immobilization. What do you mean by nutrient immobilization? So suppose this is a detritus. Suppose this is the detritus. Okay, and this detritus will be broken to form a number of small particles. Okay, so these are inorganic molecules, inorganic nutrients, right? And this is basically decomposition. This is basically decomposition, and this decomposition will be done by what? Will be done by some microbes. So suppose this is one microbe, or I can say decomposer. Okay, so these inorganic molecules, or I can say what? The minerals, or I can say the minerals which are released during the process of decomposition. Most of them, some amount will be leached out. Okay, and some amount will be uptaken by the microbe, by the decomposer. Okay, so some amount is uptaken by the decomposer. Okay, and this uptake of the minerals or nutrients by the decomposer is known as nutrient immobilization. Okay, and these nutrients will not be leached. The other those minerals which are not uptaken by the decomposer or microbe, they will be leached out with the percolating water. Okay, so this uptake of the minerals by the decomposer or microbe is known as nutrient immobilization, which prevents the leaching out of the nutrients. Okay, now let us try to understand about other topic, that is energy flow. Okay, so for all ecosystems, the ultimate source of energy is sun. Sun, except deep sea hydrothermal vents. deep sea hydrothermal ecosystems, or I can say deep sea, or I can say hydrothermal ecosystems. Sun is not the source of energy. Okay, and the flow of energy across the different trophic levels of an ecosystem is always unidirectional. Okay, and the light which is used by the plants for photosynthesis is called as par photosynthetically active radiation, and it is 400 to 700 nanometer. 400 to 700 nanometer. So the solar radiations are uptaken by the producers. They will perform photosynthesis, they will produce food, and that food will be uptaken by the herbivore, and herbivore will be consumed by the carnivores. Okay, so here you can see the energy present, solar energy transferred to the producers or uptaken by the producers. The energy present in the producers are transferred to the herbivore, and from herbivore the energy is transferred to the carnivore. Okay, so the direction of the flow of energy is from sun to the producer to the herbivore to the carnivores. Okay, now when the energy flows across the different trophic levels of an ecosystem, it follows two laws of thermodynamics. The flow of energy in the ecosystem across the various trophic level follows first law of thermodynamics and second law of thermodynamics. Okay, the first law of thermodynamics says that energy of sunlight is transferred into energy of food and heat. We know first law of thermodynamics says that energy can neither be created nor be destroyed. It will be converted from one form to another form. In the same way, the light energy is not lost. It is converted into another form of energy. Which form? In the form of food, in the form of heat. Okay, and this flow of energy across the ecosystem, it also follows second law of thermodynamics. And what does the second law of thermodynamics says? The transfer of energy is accompanied by loss of major part of food energy as heat. That means when the energy is transferred from one trophic level to another trophic level, the transfer of energy is accompanied by loss of a major part of energy, and that energy which is lost is mainly in the form of heat. Okay, and the energy which is present in the food is concentrated form, and the energy present in the heat in the form of heat is called as dispersed form of energy. Okay, then let's talk about ecological pyramids. So the concept of ecological pyramid was given by Charles Elton. That is why we call ecological pyramids as Eltonian pyramids. And in this case, we graphically represent ecological parameters. The ecological parameters can be number of organisms present in different trophic levels. That pyramid will be known as pyramid of number. That ecological parameter which we use can be total biomass present at different trophic levels. That pyramid will be known as pyramid of biomass, or that can be total energy content present at different trophic levels. That pyramid will be known as pyramid of energy. Okay, and these ecological pyramids can be upright, can be inverted, or can be spindle shaped, but in most of the cases it is upright. Okay, and whenever we draw an ecological pyramid, the first trophic level, that is the producers, they are present at the bottom side. Okay, and the higher trophic level are present at the upper side. Okay, so that means I can say, suppose these are the various trophic levels. Okay, so at the bottommost side, the trophic level will be first trophic level, then the upper trophic level will be second trophic level, third trophic level, and fourth trophic level. That means producer, consumer, and primary consumer, secondary consumer, and tertiary consumer, like this. And you can see that the width of the boxes is different. So what does this width is going to signify? So if the width is more, that means the parameter, whatever the parameter we're considering, whether number, biomass of energy, that will be more. Okay, in this trophic level. So in this because the width of the first trophic level is more, that means the quantity of the parameter will be more, and width of the second trophic level is less. That means, that is why I can say that as compared to trophic level first, the parameter will be present at second trophic level is less, like this. Okay, so let's talk about the pyramid of number. Pyramid of number can be upright, can be inverted, or can be spindle shaped. But in most of the cases it is upright in nature. So suppose let me take an ecosystem which of a grassland or pond ecosystem. Okay, so in the first trophic level, that is at the producer level, the number of species will be maximum, and when we move to the highest trophic level, the number will be minimum. Okay, that means when we move from lower trophic level, first trophic level, to the higher trophic level, the number of species keeps on decreasing, and this type of pyramid is known as upright pyramid. Okay, so what about the inverted? This is found in tree ecosystem, and spindle shape also found in tree ecosystem. So what about inverted? So let us take one tree. So let us take one tree, and on that one tree, suppose 10 birds, 10 herbivorous birds are present. Okay, and on each herbivorous birds, suppose 50-50, or suppose 30-30 parasites are present, lice are present. Okay, so 10 birds, that means 300 lice will be present. So I will say 300 lices are present. So you can see that one tree, 10 birds, and 300 lices or parasites. So this is trophic level first, this is trophic level second, and this is trophic level third. So when we are moving from lower trophic level to higher trophic level, what is happening? The number is increasing. So this is inverted. This is inverted. Okay. So here this is trophic level first, this is trophic level second, and this is trophic level third. Okay. So let me take one example here. Suppose this is one tree, and on that one tree, suppose 10 birds, 10 herbivorous birds are present. Okay, and all those 10 herbivorous birds, they are eaten by one hawk. So what happened? At the middle, at the second trophic level, the number is more, but at third and first it is less. So this type of pyramid is known as spindle pyramid. Spindle pyramid. Okay, now what about pyramid of biomass? Pyramid of biomass can be upright or can be inverted. So this is an example of what? Upright pyramid. Upright pyramid of biomass, and this is inverted pyramid of biomass. Inverted pyramid of biomass. Okay, and this upright pyramid of biomass is normally found in terrestrial ecosystem, or I can in other words I can say most of the terrestrial ecosystem, they are upright normally, and most of the aquatic ecosystems in aquatic ecosystem, the pyramid of biomass is normally inverted. Okay, so here you can see at the first trophic level, the biomass is maximum, second trophic level little lesser, third trophic level even lesser, and fourth trophic level minimum. So this is inverted. So this is upright. Okay, and suppose at first trophic level I'm taking phytoplanktons. So less number of less biomass, phytoplanktons have less biomass, and they're eaten by zooplanktons which have higher biomass. So this will be inverted. Okay, what about pyramid of energy? Pyramid of energy is always upright. Pyramid of biomass can be inverted or can be upright. Pyramid of number, it can be upright, it can be inverted, but pyramid of energy is always upright. Why? Because the flow of energy is always unidirectional. Because the flow of energy is unidirectional, and it follows 10% law. It follows 10% law. So suppose this is sun. So the amount of energy which is falling on which is available, only 1% of the energy, 1% of the solar energy is trapped by the plant. Only 1% energy. Okay, and this will be trophic level first, this will be trophic level second, this is trophic level third, and this is trophic level 4. Okay, so the energy, so only 1% of the energy available to the plants is absorbed by used by the trophic producers or plants. Okay, from trophic level first to second, only 10% energy is transferred. From trophic level second to the trophic level second to third, only 10% energy is transferred, and from trophic level third to fourth, only 10% energy is transferred. That means across different trophic levels, only 10% energy is transferred, and this is called as 10% law, which was given by Lindemann. Okay, so only 10% energy is transferred, and this law is known as 10% law. Okay, and this was given by Lindemann. This was given by Lindemann. This is very, very important. Okay, this is 1% and this is 10%. Okay, that means when we move from lower trophic level to higher trophic level, the the amount of energy present in that trophic level will be keep on decreasing. It will keep on decreasing. Okay, so what about food chain? Food chain is basically sequence of living organisms in which there will be transfer of food energy. Sequence of living organisms in which there will be transfer of food energy, and that food energy will be transferred from the producers to the primary consumer, then primary consumer to secondary consumer to tertiary consumer, like, and so on. That means I can say this is the basically transfer of food energy from producers through a series of organisms with repeated eating and being eaten. That is food chain. Okay, and each level of the food chain is known as food trophic level. Is known as trophic level, and the food chain is of three types: grazing food chain, detritus food chain, and parasitic food chain. Okay, and in aquatic ecosystem, the flow of energy, the flow of food energy mainly takes place through GFC, and in terrestrial ecosystem, maximum amount of flow of energy, flow of food energy takes place through DFC. That means detritus food chain. Okay, so the grazing food chain starts with producers only. It always starts with producers, and for this grazing food chain, the ultimate source of energy is sun. Okay, so it starts with the producers. So the example is grass. Grass is eaten by grasshopper. Okay, grasshopper are eaten by the frogs, and frogs are eaten by snake, and snakes are eaten by eagle. So this is an example of what? GFC. Okay, if I take the example of pond ecosystem, so I can say phytoplanktons eaten by zooplanktons. Zooplanktons are eaten by small fishes. Small fishes are eaten by large fishes, and large fishes are eaten by some other organisms like eagle or etc. Okay, what about DFC? DFC never starts with the producers. It starts with the detritus. So what is detritus? This is basically dead organic matter. Okay, and this detritus is eaten by what? A detrivore. Example is earthworm. Earthworm is eaten by sparrow, and sparrow is eaten by falcon. Okay, what about parasitic food chain? Parasitic food chain, I can say suppose there is a plant. Okay, there is a plant, and this plant is host plant. Okay, and on this host plant, parasitic birds are there. Okay, or I can say herbivorous birds are there. And on these herbivorous birds, some parasites are present like lice and bugs. So this is parasitic food chain. So these birds are parasitic to the plants, and these lice and bugs are parasitic to the birds. Okay, so this is food chain. And what about food web? Naturally, food chains do not exist. Naturally, food web exists. So what is food web actually? Food web is actually a interwoven food chains. That is food web. Okay, and food chain assumes that a given organism will occupy a specific trophic level, but it is incorrect. A given organism can operate at different trophic levels. So if I eat some vegetables, I'm operating at trophic level 2nd. But if I eat, suppose if I'm eating for example chicken, so suppose if I'm eating some herbivorous birds, I will be operating at a 3rd trophic level. So a given organism can operate at different trophic levels as per food web, but as per food chain, a given organism will operate at only 1 trophic level, which is incorrect. Okay, so the food web and the at which trophic level the organism will operate, that will depend upon the food preference or the food availability for that organism. Okay, now let us talk about the last part of this chapter, and that is second last part of this chapter, and that is ecological succession. So what do you mean by ecological succession? Suppose there is an area, in that area there was no plant, no animal. Okay, and in that area gradually some plants started to colonize, and ultimately forest developed. And that process in which an area which had no plants, uh, the forest developed, that process is called as ecological succession. Okay, and this process is very slow process, and in this process we can predict which species will first come, and then which species will come, and which species will come. We can predict that one. So that is why I can say this ecological succession is gradual and fairly predictable changes in species composition of a given area. Okay, and where does this ecological succession take place? Based on that, we can classify ecological succession as hydrosphere and hydrarch and zedarch. If the ecological succession takes place in aquatic ecosystem, that will be hydrarch or hydrosphere. If it takes place on terrestrial conditions like rock or sand, then we can call that as zedarch or xerosphere. Okay, so the example of zedarch. So if I take the example of what ecological succession on rock, so the first organisms or the first organisms which colonize will be lichens. Okay, and these lichens are called as pioneer community, because the first organisms or the first species which colonize during ecological succession, they are called as pioneer community. So in this case, the pioneer community are lichen. The pioneer community are lichen. Okay, and when the lichen come and colonize on the rock, they will produce little amount of soil, and that soil will be sufficient to support some larger plants like bryophytes. And bryophytes have liverwort, hornwort, and mosses. So here the organisms which are going to colonize are basically mosses. Okay, and the mosses will further develop more amount of soil, and that will support even larger plants, and that is why now the herbs will come and grow. Okay, and the herbs will produce more, will make the habitat more suitable for even larger plants like shrubs, and these shrubs will make that habitat more suitable for even larger plants like trees. Okay, so at the end I will get the trees, and these trees or the forest is now called as climax community. So the first community which develops during ecological succession is pioneer community. Okay, and the last community is called as climax community. And this climax community is more or less at equilibrium state. Okay, and this sequence you have to remember. What about the hydrarch then? So you have to remember over here, in hydrarch or hydrosphere, the pioneer community is plankton, phytoplankton, and the climax community is forest. So this one is forest. This one is forest. This one is forest. Okay, so the first community or the pioneer community is phytoplankton. The second community is submerged plant stage. Okay, third is submerged floating stage. This is phytoplankton. This one is submerged plant stage. This one is submerged free floating plant stage, and this one is reed swamp, and this one is marsh meadow stage, and this one is scrub stage, and the final one is forest. Okay, and you have to remember this sequence order. Okay, and you can clearly see that in whether it is hydrarch or zedarch, the climax community is forest. Zedarch forest, even in the hydrarch it is the forest. That means in all types of ecological succession, the climax community is same, that is forest. But the pioneer community is different. In case of hydrarch, it is the pioneer community is phytoplankton. In case of zedarch, for example, on rock, it is lichen. Okay, and on rock water content is very less. That means dry condition is there. Okay, and in forest the soil will have medium amount of water, moderate amount of water, and that condition will be mesic condition. So in zedarch, what is happening in zedarch? The dry condition is being converted into the mesic condition. Okay, dry means no water or very less water. Mesic means moderate amount of water. Okay, and in this case, uh, the ecological succession is taking place in the water body. So here, in hydroc, the water is present in maximum amount initially. So I will say water rich condition is there. Initially the water rich condition is there, and finally I am getting the forest, and in the forest medium amount of water is present. That means basic condition will develop. That means I can say whether that is hydroc or xeroc, the final condition will be basic. Okay, in hydroc the initial condition will be water rich condition, in xeroc it will be dry condition. Okay, and during the process of ecological succession, what happens? The plant community, the species present in the plant community will keep on changing, and accordingly the decomposers and the animals will also change in that area. Okay, and during the process of ecological succession, the species composition changes. Some species will be completely disappeared, some will some number will decrease, and some number will increase. Okay, so this is ecological succession. Clear? So suppose there is an area, in that area there was no plant. So in that area, if some gradually the species come, different species come and they start to colonize, and ultimately there a forest is formed, that process will be called as ecological succession. That based on the nature of the habitat where the ecological succession is taking place, we can classify that as xeroc or hydroc. Okay, now the last part, that is the nutrient cycle. And this is the fourth functional aspect of the ecosystem. So I'm writing here nutrient cycle. So what are the functional aspects of ecosystem? Productivity, decomposition, energy flow, and fourth one is the nutrient cycle. Okay, what about structural aspects? Species composition, stratification. Okay, so nutrient cycle takes place between living organisms and abiotic component like water, air, or I can say or rock. Okay, so this nutrient cycle takes place between living organisms okay, and living organisms and other parts, like it can be water, or it can be air, or it can be rock. Okay, so some nutrient will be transferred from living organism to these components, or from this component to the living organism. So this is basically nutrient cycling. Okay, so and because this is taking place between living organisms, so I will call this as bio. Okay, and all these three things can be considered as geo. So biogeochemical cycle, because the nutrients are basically chemical, so we call that as biogeochemical cycle. Okay, just a minute. Now based on the source reservoir pool, this nutrient cycle can be classified into two groups. Reservoir pool means the source of the nutrient, or the source of the nutrient. We call that as what? Reservoir pool. Okay, so based on the source of reservoir pool, the nutrient cycle is of two types. One is called as gaseous cycle. Okay, and second one is called as sedimentary cycle. Okay, and in the first case, the reservoir pool of the nutrient is air, and in the second case the reservoir pool is rock. Okay, so if the reservoir pool of the nutrient is air, that is gaseous cycle. If it is rock, it is sedimentary cycle. Examples I can take here: carbon, hydrogen, oxygen, nitrogen. Here I can take the example of what? Sulfur and phosphorus. And one important point is that the sulfur shows both gaseous cycle and sedimentary cycle, both. Both types of cycles are observed, but it is mostly sedimentary cycle. Okay, so let's talk about the carbon cycle here. Okay, why carbon is important? Because living organisms are made up of biomolecules, and every biomolecule is made up of carbon. That is why the carbon is very, very essential. Okay, so we know that these are the plants, and these plants, they use the sunlight. Okay, and they will uptake the CO2 which is present in the atmosphere. Okay, and they will synthesize the food, and that food will be uptaken. That food will be uptaken by the what different types of organisms, like for example, the grasses will be uptaken by the grazing animals. Okay, and these animals will release CO2. They will release CO2 during the process of respiration. Okay, and these animal, whether that is animal or plants, when they undergo when they become dead and decay, that carbon will that carbon will be released, will be stored in the form of fossil or fuels, and they will be used by different in different areas, different areas like factories or automobiles, etc. Okay, and burning of those fuels or fossils or fuels, what will happen? Not fossils, no, burning of the fuels that will release CO2. Okay, and the CO2 will come back to the atmosphere. Okay, so this is very simple. Okay, now what about the phosphorus cycle? Phosphorus is very important. Why? Because this is a component of cell membrane. How? Because one of the major lipids of the membrane is phospholipid, and phospholipid has phosphorus. And this is also a component of what? Nucleic acid. Nucleotide has phosphate, so phosphorus will be there, and that is why this phosphorus is very, very important. Okay, so the from producers, the phosphate is uptaken by the consumers. Okay, and the consumers, when they become dead, they will produce that will become detritus. Even the producers, when their leaves or the leaves will fall, or when the producers will die, they will form the detritus. Okay, and this detritus will be decomposed, and the phosphorus will be released into the soil. The soil and the phosphorus will be present in the soil solution. Okay, and the producers, just a minute. Okay, now and in the soil solution, from the soil solution, the producers will uptake the phosphorus in the form of phosphate. Okay, and in the soil solution, the phosphorus is released from the rock. That means I can say from rock and detritus, the phosphate is added to the soil solution. Okay, and from soil solution, the phosphorus is uptaken by the producers. Okay, and from the phosphorus is uptaken by the consumers. Okay, I repeat here, this is the last part. So in the soil solution, phosphorus is present, and the phosphorus is added in the soil solution by the rock and by the detritus. Okay, detritus will add the phosphorus by the process of decomposition, and the rock will add the phosphorus by the process of weathering. Okay, and the phosphorus in the soil solution now will be uptaken by the producers. From the producers, it will be uptaken by the consumers. Whether that is producer or consumer, when they become dead, that will produce the detritus. So this is how the phosphorus cycle takes place. Okay, so this was all about the ecosystem. Okay, so let me revise in a minute. So ecosystem is basically the functional unit of living world, which has both biotic and abiotic component. Abiotic components can be classified into various groups like edaphic factors, okay, climatic factors, topographic factors. And biotic component are of various types. They can be classified into various groups like producers, consumers, detrivores, decomposers, scavengers, etc., parasites, etc., etc. And producers are known as converters or transducers. Why? Because they convert one form of energy into another form of energy. And the primary consumers, that is the herbivores, they are called as key industry animal because they convert plant material into animal material. Okay, and the function, when I talk about the aspects of ecosystem, I have two aspects of ecosystem: structural and functional. Structural aspect, the examples of structural aspects are species composition and stratification. When I say species composition, means in an ecosystem what type of species are there and how many different types of species are there. What is stratification? The vertical distribution of different species in an ecosystem, that is stratification. What are the functional aspects of ecosystem? Productivity, decomposition, nutrient cycling, energy flow. Okay, energy flow always takes place across the different trophic levels by following 10% law. Okay, productivity is nothing but the rate of biomass production. Okay, rate of biomass production per unit area. So if the productivity is due to producers, it is primary productivity. If it is by the consumers, it is called as secondary productivity, and if it is by the community, that will be called as community productivity. Okay, and the primary productivity is again of two types: GPP, NPP. And NPP is basically GPP minus respiration, and NPP is basically available to the decomposers and the consumers. Decomposition is equally important as the productivity is important, and decomposition is done by or carried out by decomposers and detrivores, which includes 4 different events: fragmentation, which is also fragmentation, then mineralization, humification, and leaching. Okay, and what about the ecological parameters? Ecological pyramids. This is the graphical representation of various ecological parameters at different trophic levels. They can be upright, they can be inverted, they can be spindle shaped, but in most of the ecosystems, most of the ecosystems, the ecological parameters are upright in nature. Okay, so this was all about. And what is ecological succession? This is basically a predictable change or gradual change which takes place in an area where there was no plant, and finally the forest will develop. Okay, and based on the habitat, we classify that into aquatic, into hydroc or hydrosphere, and xerac or xerosphere. Okay, and during the process of ecological succession, species composition, species diversity changes, and the nature of scavengers, nature of decomposers, and nature of animals also keeps on changing, and the total biomass increases, total biodiversity increases, and from and the ultimate condition will be magic condition, either from dry condition or from the water rich condition. Okay, finally the nutrient cycling. The nutrient cycle can be of two types: gaseous or sedimentary cycle. Okay, so this was all about ecosystem. Thank you.